I took the cap off the pen, not the cap off my water, and I'm like, at least I didn't try and drink the pen. That would have been bad. I'm already all like swollen and bruised. Okay, so we have two different types of regulatory systems that control the body. The nervous system and the endocrine system. And we've essentially talked about all of this already because we talked about synaptic communication and we talked about endocrine communication. But we'll just kind of apply it really fast. So the nervous system obviously regulates the body. We looked at that last semester and we saw that it controls a lot, right? The nervous system controls like glands or some of our glands, like sweat glands. It controls skeletal muscles, sensation, right, is via the nervous system. That's how we know what it is that we're touching or smelling or seeing or hearing. So we talked about the nervous system quite a bit. The nervous system utilizes what kind of communication? Synaptic communication. So all the stuff we just learned about synaptic communication applies to the nervous system. One, it utilizes neurotransmitters, and those neurotransmitters are released very close to their target. So that means it's a very, very, what kind of effect? Narrow or widespread? Narrow. A very, very narrow effect. Right? You only affect that one intended target. You don't send those neurotransmitters all over the body. Typical targets of the nervous system include muscles. We talk about that. Some glands. And then also neurons, right? Neurons intercommunicate with each other very frequently, especially in the CNS. The nervous system is a very, very fast way to regulate the body. Very, very fast. Okay? And the effect is very, very brief. It happens and then it's done. Just like that. So we utilize it to manage short-term stresses. Again, stuff like muscle movements. Things like sensation. This has to be rapid. There is no point in vision if it takes 5 minutes to process what you're seeing, or hearing, or tasting, or touching, right? Imagine I step on a thorn, it goes in my foot, that hurts, so my muscles contract and I remove my foot. That has to be quick, right? If I stepped on that thorn and it took 10 minutes for the hormones to be released and for them to come up and for them to tell my brain, hey, that hurts, that doesn't feel very good, and then I make another hormone and I release it to the bloodstream and it sends a signal out to all of my muscles to say contract, and then all of my muscles contract 5 minutes later, like that's useless, right? I need to tell just this muscle to contract and I need it to happen fast because that hurts. So it's very, very quick, very, very narrow, specific response, and it goes away fast. Endocrine communication, which is really what we're going to focus on for the rest of the time today. Endocrine communication, the endocrine system utilizes what kind of communication? Endocrine, what do you know? And endocrine communication utilizes hormones. Hormones travel through what? They travel through the blood. So they act far from their site of release. And what does that mean as far as their effect? Widespread or narrow? Widespread. So they have a very widespread effect. Targets include pretty much any body cell. Every one of most of your body cells can be affected by one hormone or another. Hormones really work all over the body. Now they take a while to work. Again, just because of what they do. They're released, they enter the bloodstream, they have to travel through the bloodstream, leave the bloodstream, go to a target, bind to a receptor, and cause something to happen. Turn on a channel, turn off a channel, activate a gene, increase metabolism. Like that is not a millisecond kind of a thing. That takes time. Even just the getting into the bloodstream and traveling around the body, like that takes a little bit of time. So it takes minutes, or sometimes more, hours, for endocrine effects to start taking effect. But they last a long time, okay? And this could be minutes that they last, this could be hours, this could be days. The effects of endocrine regulation last a very long time. So we utilize them for long-term things, long-term processes. We said growth, right? Growth of the body, that's an ongoing thing. That is not like a rapid one and done. Metabolism, development, and lots of long-term processes utilize endocrine regulation. So again, from here on out in the chapter, now that you kind of have a little bit of a basis, we're going to just focus on endocrine communication. The endocrine system, again, utilizes hormones. When we look at hormones, we can kind of break them up or classify hormones into a few different categories or types based on their structure. So we're going to look at their actual chemical structure and the properties of those structures in order to determine what type of hormone it is. Now, I will tell you, different textbooks break these up in different ways. Some break them up into 3 categories, some break them up into 4 categories. I'm just going with the latest version of the textbook and breaking it up the way that it does. But the key principles will apply regardless of how many groups you classify them as. So, amino acid derivatives are relatively small hormones that come from individual amino acids. So we are not linking together a bunch of amino acids to make a really huge molecule here. We're essentially taking one amino acid and we're adding a few things to it. We put a little bit here, a little bit here, a little bit here, but the hormone is really based off of one amino acid. We look at hormones that are based off of tyrosine and hormones that are based off of tryptophan for the most part. Tryptophan ends up giving us a hormone called melatonin, which we'll see is important for sleep cycles. The thyroid hormones, T3 and T4, are based off of the amino acid tyrosine. Most amino acid derivatives, so like epinephrine and norepinephrine, could also fall into this category when we use them as hormones, not as neurotransmitters. But they would also fall into the category of amino acid derivatives. So again, they're relatively small. When we look at the amino acid derivatives, for the most part, we will say that they are hydrophilic. So for the most part, they like water. They're relatively small. The only thing here that's going to be different is the thyroid hormones. Okay, and we'll talk about why that's important later when we talk about protein binding and where they act and all of that. It's going to be important. But the thyroid hormones are the only amino acid derivative that's odd, that is not hydrophilic. And lipophilic, or you could say not hydrophilic. Again, the fact that it's not hydrophilic is going to be important for the way that it travels through the blood. So those are amino acid derivatives. What are peptides, just in general, with a peptide? A bunch of them, right? You start to put amino acids together and you make a peptide. Like remember we call a protein a polypeptide? So there's a bunch of peptide bonds, a bunch of amino acids linked together. So peptide hormones are bigger than amino acid derivatives because now we don't have just one amino acid, we've got multiple. And we could link together 8 amino acids, or we could link together 200 amino acids. Okay, we've got all different kind of sizes here, and this is where some books will break this up, and they'll say peptide hormones versus proteins, and that's just kind of based on structure or how many amino acids are there. But for our purposes, we'll stick with peptide hormones, and again, we've just got chains of amino acids. This is the most common type of hormone. Most of our hormones that we'll look at are peptide hormones. So things like insulin, TSH is thyroid stimulating hormone. ADH, we've already talked about. Do you guys remember what is ADH? Good job, guys. Antidiuretic hormone. And GH is just growth hormone. So peptide hormones, again, they're the most common. They're big because they're chains of amino acids. They're relatively polar molecules, so that means that they are hydrophilic. They're hydrophilic, so they like water. This tells us a couple things about them. One, they're soluble in the blood because the blood is mostly what? Water. And remember, likes dissolve likes. So like oil and water don't mix, right? They don't like each other. But water and anything else that's hydrophilic are fine. I can put peptide hormones in water and they're soluble. They spread out nicely, they interact with the water, they're perfectly fine. Also, this tells us that they're going to bind to extracellular receptors on their target cell. When we look at a target cell, and we've got a little hormone here, and the hormone's going to come to the cell and cause something to happen. It can bind to a receptor out here, an extracellular receptor, or if that hormone is able to enter into the cell in some way, it can bind to an intracellular receptor. In a minute, like or towards the end, we'll see that we've got intracellular receptors on the nucleus, on the mitochondria. We've got them in numerous different areas inside the cell. But in order to bind intracellularly, the hormone has to get inside the cell. And when we look at peptide hormones, they're big and they're hydrophilic. So they don't get in the cell. So they bind to extracellular receptors. So the last class that we'll look at includes the lipid derivatives. When we look at lipid derivatives, they all have one kind of common structural feature, and that is that they have 4 carbon rings. Okay, all of the lipid derivatives that we'll look at have a base structure of 4 carbon rings. So you remember when we did the chemistry chapter, we looked at steroids, right? And we said steroids were lipids. Steroids are lipid derivative hormones, and they've got those 4 carbon rings. All of our lipid derivatives will have these carbon rings, and that makes them very lipophilic, right? Those rings are non-polar, so they're lipophilic. What would you say if you were saying it in terms of their water, if they like or don't like water? Hydro what? Phobic. They are hydrophobic. They do not like water. We see that they're made from fatty acids or cholesterol, which, well, you know, are also examples of our lipids when we talked about the lipids in the chem chapter. And when we look at these lipid derivatives, we call them steroids. So if you've heard any, I mean, I'm sure you've heard the term steroids before. That's telling you that these are lipid hormones. So things like androgens. What's the most well-known androgen? Testosterone. Okay, androgens like testosterone, estrogens and progesterones, which are obviously more common in the female. Corticosteroids like cortisol or hydrocortisone, aldosterone, and we'll talk through all of these, like as we go through the different hormones and different glands, we'll talk through each of these in detail. But the point is, these are all steroid hormones. So they all have these 4 carbon rings. That means they're all hydrophobic. They do not like water. Now, the reason that this is important that they're hydrophobic is because they're insoluble in the bloodstream. Again, likes dissolve likes. The blood is water. So things that like water will dissolve in it. These are not like water. These are fats. So just like when you put oil in water, the oil all clumps together, right? That's what these hormones try to do. When you put them in the bloodstream, they're not soluble. They don't spread out and flow nicely. They try to all clump together. And we obviously can't have that happen, right? We don't want these big, huge drops of fatty hormones going through our bloodstream. So we need to do something about that. And we'll see that we utilize protein binding, or carrier proteins, in order to make them more soluble in the bloodstream. Now, because they are lipophilic, because they like lipids, they bind to intracellular receptors. They can go through that plasma membrane. They don't mind that lipid bilayer. And they can actually enter into the cell and bind inside the cell. Okay, we'll see that with the thyroid hormones as well, that they also bind intracellularly. So, hormones, we said, travel through what to get to their target? The blood, and the blood is mostly water. So as targets travel through the bloodstream to get to their target, they travel either free, meaning they don't have anything attached to them, or they travel bound to some sort of a carrier protein. Whether or not they travel free or bound depends on their solubility in the bloodstream. Okay? If they're not soluble in the blood, we need to make them soluble in the blood. We do that with the carrier protein. If they're already soluble in the blood, then we don't need to adjust anything. They don't need a protein. They're already going to travel through the bloodstream fine. So, free hormones. Again, these are hormones that do not have a what? A carrier protein. They travel by themselves through the bloodstream. These ones are the ones that are soluble in the bloodstream. Okay, so most amino acid derivatives, all of the peptide hormones, right? So the majority of our hormones really, but like melatonin, all of the peptide hormones that we'll cover, all of those are soluble in the bloodstream. When we look at the free hormones, we see that a key difference between them and the bound hormones are that they have a shorter duration of action. So any of our free hormones, once the gland releases them into the bloodstream, they're going to be in the bloodstream and doing something for less than an hour. Then the effects that they have on the cell can last longer. You know, once a cell starts making enzymes or channels or whatever, like that can keep continuing. But the enzyme or the hormone is traveling through the bloodstream and affecting targets for about an hour or less. Now, what happens? Like how does the hormone stop working? How does it get inactivated? There's a few different things that can happen. One, it can go work. Okay, so I have all of these hormones in the blood, and I said they're going to be there for about an hour. Some things that happen are the hormone will leave the bloodstream and go act on its target cell. Right? It's no longer in the bloodstream anymore. It's done what it needs to do. It acts, it gets destroyed, whatever, it's gone. Also, they can be broken down in the liver or excreted in the kidneys. Okay, if this blood goes to the liver before this hormone has ever gotten a chance to do anything, the liver has enzymes that will metabolize it, right? That will break it down and get rid of it. Now it's gone. Same thing can happen in the kidneys. If this blood gets to the kidneys before the hormone's ever acted, the kidneys can filter it out, put it in the urine, and it can go. They can also be broken down by enzymes in the plasma or interstitial fluid. Okay, so we do have enzymes that could start breaking it down here before it does anything, and then in the interstitial fluid as well. Okay, so those are the three things that happen. Either it gets broken down, it gets taken out by the liver or kidneys, or it actually works. But the point is, if it's a free hormone with no plasma, with no carrier protein, once you release into the bloodstream, you've got less than an hour, and then it's all gone. It's out of the bloodstream. Bound hormones are called bound because they travel through the bloodstream bound to something. They are not free by themselves in the blood. They have a carrier protein. Why? Why did we say they need a carrier protein? It increases their solubility, right? So most of these bound hormones are lipid-soluble hormones. So things like the steroids, testosterone, estrogen, progesterone, corticosteroids, hydrocortisone, all of those steroids, because we just said they're lipids. Lipids don't like water. They're going to clump together. We want them to dissolve nicely. So we add a carrier protein to each one. Now that all of a sudden allows it to interact with the water and be fine. Also, we said the thyroid hormones. Which we'll see are T3 and T4. Those utilize carrier proteins as well. Now, the carrier protein, it's there for solubility to make the hormone soluble in the blood, but what it ends up doing also is making it active for much, much longer. So the reason for this is that when the kidneys are, well, there's kind of twofold, I'll talk through this, but one is when the kidneys are filtering stuff out of the blood and getting rid of it in the urine. That's based on size. So if I have, and the ability to cross membranes. So if I have a relatively small lipid-soluble thing that can just cross membranes easily, it very easily gets pushed out into the urine and we lose it. If I have a really small lipid-soluble thing, and what I do instead is attach a big, giant protein to it, now all of a sudden it can't get pushed out anymore. These carrier proteins are really big. It makes that hormone so much bigger that now it can't get filtered out of the kidneys any longer. They are plasma proteins. Large proteins do not get filtered into the urine. So if we put a carrier protein on a hormone, it cannot be lost in the urine any longer. Okay, so that means we have so much more hormone present in the bloodstream for a lot longer period of time. What we see is that we have an equilibrium state that exists in the blood between the free hormone and the bound hormone. So say I have 1, 2, 3, 4, 5, 6. So 6 units of the hormone, and 2 of them are free, and 4 are bound. So like say that's my equilibrium state. 2 free to 4 bound. And then this free hormone is the only hormone that can actually leave the bloodstream and go act on a target cell. Again, the carrier protein's huge, so that can't, remember, plasma proteins don't get filtered out of the capillaries, right? They stay in the bloodstream. So when this carrier protein is present on the hormone, it cannot leave and go act on the target cell. It's essentially just stuck right there in the bloodstream. So these ones right here, these two, one of them can say leave and come here and act on the target cell. But now I've disrupted that equilibrium. So when I take one of the free ones out of the bloodstream, one of these carrier proteins will release another hormone. So now I've got another one ready. Right? All of these are protected. They're not going to get filtered out of the kidneys. They're not going to get metabolized. They're not going to go act and get metabolized out here. They're just hanging out, right? It's like a little bank account of hormones just hanging out in the bloodstream with their carrier proteins that protect them. And then if, say, this hormone leaves and goes and acts, then all of a sudden I've messed up my equilibrium. So then one more of them will say, okay, there you go, I'll free up. Carrier protein goes away, and now we've got another one that's ready to act. Right? And that happens and happens and happens. But all of this time, we have all of these other ones that have their carrier protein that are being protected. If they didn't have that carrier protein, they'd go to the kidneys and they'd be gone. We'd filter them out into the urine and they'd be gone, or they'd go get metabolized in the liver. But that carrier protein protects them. And then again, it just kind of, every time we lose a free hormone and we need more, it'll just kind of release it, and release it, and release it. So these are active for a much longer period of time. They have a really, really long duration of action because of that carrier protein. So for example, like in patients who have really severe hyperthyroidism, one of the treatments that we'll do is to completely irradiate and destroy the thyroid gland. But after you destroy the thyroid gland, it's not instantaneous that the patient's fine and they're fine, the thyroid hormone's all gone. They actually have really high levels of thyroid hormone continuously for over a week after that, because they had so much thyroid hormone bound to carrier protein already in their bloodstream. So you can completely take out the thyroid gland, but they still have thyroid hormone present for 7 days later because of all of it that was already bound to the carrier protein. It takes so long to actually free that up and get rid of it.